drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. Eugene Chang, thank you for joining me.
It's a pleasure.
Can you start off by just telling everyone a little bit about who you are and what your lab studies?
Sure. So I'm a physician scientist at the University of Chicago, a professor of medicine. And my subspecialty training is in gastroenterology. And that's how I got interested in this new vital organ that we've discovered, which is the gut microbiome. And this is something that for years we always thought was, something that we had to avoid, and it was a cause of disease. But I think it now is realized that this is a organ system that plays a tremendous role in maintaining our health. We absolutely require our gut microbiome, for instance, to develop metabolic as well as immune processes. And when things go wrong, like any other vital organ of the body, a lot of diseases and consequences will occur.
Mm-hmm, mm-hmm. So you use the term vital organ. I think you've described the microbiome as an acquired organ. What exactly does this mean? And in what sense is the microbiome an organ, like a traditional organ that we would think of?
Well, let me give you some examples. If you damage the microbiome or eradicate it, let's say with a cocktail of broad spectrum antibiotics, which is common. We see this in clinical practice. um, patients become prone to this uh, pretty terrible disease called Clostridioides difficile colitis. We see a lot of it at our medical center, and I'm sure others elsewhere see it as well. This is when you lose your gut microbiome through the antibiotics, it renders your gut susceptible to these opportunistic infections. In this case, something called C. difficile. And this causes, in many patients, a severe colitis. And the treatment of choice is to add more antibiotics to try to kill the C. difficile, but at the same time, you're damaging even further the commensal population or the healthy microbiome. The other reason for this is that it can be transplanted like other organ systems. We have this method called fecal microbial transplant, where we take somebody's colonic microbiome and transplant it into individuals that may have, let's say, C. difficile colitis, or in other instances, people are using FMT for things like inflammatory bowel diseases, even metabolic disorders. So this is also associated with rejection of the donor microbiome in some individuals. So in many ways, our microbiome is a organ of our body. It provides many beneficial functions that we have to have day to day. And if it's damaged, like any other organ of the body, there are consequences. And we acquire it through actually early in life. Most of the early life microbiome comes from our mothers through vaginal delivery. And over time, we acquire other microbes, but the microbes we acquire through life are very specific to individuals. Your microbiome is going to be very different from my microbiome. And that's because your genetics and your biology have selected, and this is not stochastic, it selects the kind of microbes that fit and are needed by your body. So these are things that I think really help us define the gut microbiome as a vital organ.
Mm-hmm, mm-hmm. So, unlike a normal organ, it doesn't have a defined border. This is sort of a diffuse thing. If we were to consider a normal organ like the liver, it's got a defined place that it exists. It has borders. You can point to it. You can pick it up and hold it if you were to remove it surgically. But functionally speaking, this really is like an organ system in that it's doing something. It's changing over the course of development. You can transplant it from one individual to another with consequences. That transplant may or may not take. It may be rejected depending on how closely it matches the original organ, so to speak, that it's replacing. And in functional terms, it's if we analogize it to the liver, say, it's doing things, chemically speaking, just like a traditional organ would. It's transforming food and other items that go into the body. The expressional level of the enzymes and things that it uses to do this are changing over time. It can become bigger, smaller. It can change its composition. Its cells can turn over and all this stuff. So even though you can't see it and point to it, and it's this more diffuse thing, it really can be thought of as like an organ, just like any other.
Yes, I think you summarized it very well.
And when we start to think about what the microbiome is doing and how it can be hurt or helped, you mentioned antibiotics, and antibiotics are widely used today for, sometimes rightly so. Sometimes people have severe infections that are bacterial. Antibiotics will help kill the bug that's causing the infection. Sometimes, when these things are bad enough, the body can't do this on its own, and antibiotics definitely have been a useful tool throughout history. On the flip side, antibiotics are so widely used and have been for so long now that they've often been overused or misused. People get antibiotics even when they don't have a bacterial infection. And as you mentioned, the antibiotics, even when they're used right, and they work against a particular bug that is causing a problem, they're never totally specific. An antibiotic is never going to be specific for one species of bacteria. And even though you can hurt the pathogens in your body with antibiotics that you want to get rid of, you're also going to hurt the microbes that you want in your body that are part of this so-called organ system called the microbiome. Can you give us a sense for the general effect that antibiotics have had on microbial health in our bodies in the modern world?
Well, so what you say is correct. They have a purpose, so we have to be careful in using them. There has to be a clear indication. We can't take it like candy. Um, and I think that the consequence is that sort of the dark side of these antibiotics is if they're used for an extended period, it causes considerable collateral damage to your healthy microbiome. And in most cases, if let's say the treatment with antibiotics is just a few weeks, the rest of your microbiome is resilient enough that it can bounce back. But over time, extended weeks to months, you will extinguish key populations of your healthy gut microbiome. That's when you get into real big trouble. Now, to bring this to sort of talk about the scale of how we damage our microbiome, it's not just with antibiotics. Western diets, which are usually high in fat, low in fiber, very calorie dense, really damage the microbiome. I think this is very evident that the microbiome of our population in the United States is quite different from, let's say, microbiome from somebody from Africa or South America. And this probably accounts for the emergence of diseases that we never really saw very much of 200 years ago. An example of that is inflammatory bowel diseases. The first case of inflammatory bowel disease was actually described in the mid 1800s. Prior to that, we didn't know that it existed. So over that period of time, which is only 200 years, that's very short on an evolutionary scale. So this is something that we have done to ourselves. We've changed our microbiomes. That has sort of disrupted this host microbe interaction, this co-evolution of us with our gut microbes in a way that then makes some of us, particularly those that have some genetic susceptibility, prone to the development of these new age disorders. So other examples include asthma, I think, and certain allergies, food allergies, pollen allergy, even metabolic disorders like obesity and diabetes, fatty liver disease are probably due to this, the change of our gut microbiome that disrupts the normal physiological relationship between our microbes and ourselves.
Mm-hmm, mm-hmm. I want to talk in general terms to start out with the ways in which the microbiome can be changed to become less healthy or more healthy, broadly speaking. There's different things we can talk about here, but two aspects of the microbiome, two features that I think are related to each other and to how healthy the microbiome is are its taxonomic diversity, how many different species are present and which species are present, and its metabolic breadth, the types of metabolic change that set of diversity in the gut can perform or elsewhere on the body. But, assuming we're talking about the gut microbiome, you've mentioned obviously that antibiotics can affect this, diet can affect this. Modern diets and the overuse of antibiotics have in general probably made our microbiomes less good at making us healthy. The term microbiome dysbiosis often comes up in the literature. What exactly is this, and how do we think about it on a high level in terms of how it works and the types of things that induce this dysbiosis?
Yeah, so that's a good question. People have been trying to define dysbiosis. Uh, at a high level, dysbiosis means that there's an imbalance or damage of the gut microbiome. Now, how we define that in terms of scientific or clinical terms has been a challenge. Now, people use diversity. This is, how many different types of microbes you have as a one definition for eubiosis, a healthy microbiome. And when you lose that diversity, you people define that as dysbiosis. However, when you look at some of these population studies, for example, the NIH had a big program called the Human Microbiome Project. This was about 20 years ago. And what they found was that diversity actually is not a measure of health of a healthy microbiome because they studied hundreds of healthy adult subjects. And when you looked at the diversity, it ranged from very low to very high. And yet all of these individuals were healthy. So diversity really isn't a good definition. It's really based off of taxonomical profiling. Who is there?
Mm-hmm, mm-hmm. This
And
sounds like the type of thing too that's it's always going to be relative to it's always going to be based on a state of relative health and unhealth or disease. If there's many ways to be healthy and there's many ways to be unhealthy, and I think what you're getting at here is I can become less healthy than I am today. I could develop IBS or some other condition, and that may or may not involve the diversity of my microbiome going up or down. And so this is sort of a relative term here. There's no absolute one size fits all healthy microbiome, just like there's no one way to just be healthy generally.
yes, well, to that's true to some extent. So what our group is doing now is trying to define eubiosis and dysbiosis using functional measures.
Mm-hmm.
Okay, so what do I mean by that? Well, your microbiome provides critical functions. For example, what we call fermentation. This is where, when we eat dietary fiber, we don't have the enzymes to break it down into nutrients. Okay, that's done by our gut microbes. All right, so that's a very, very important function that the gut microbiome provides to us. It's also the gut microbiome provides functions like amino acid or vitamin biosynthesis, right? And it's really important in the processing of what we make. These are called bile acids. Bile acids are made in the liver, but they are then further processed and modified by our gut microbes. And that has to be done in a proper way. If something perturbs that natural process, you make these bile acid metabolites that could be cancer promoting or pro inflammatory. So, this is why we define the health of the microbiome by functional markers. We look at things like fermentation, bile acid, metabolism, and also things like vitamin and amino acid biosynthesis.
Mm-hmm.
And so we have developed a panel that we're actually submitting the manuscript today that showcases a panel we created, which was created from what we called a supervised machine learning platform. So these are ways where we can look at and screen for particular metabolites that are either made or derived by gut microbes. And that gives us a very good picture of how whether the microbiome is functioning correctly or not.
Mhm.
And on top of that, we have been able to identify those metabolites which have uh, are shared among all healthy individuals, so that we can develop these normal reference ranges.
Mhm.
Just like if you look at uh, the clinical medicine as a whole, we depend on these types of reference ranges. Blood glucose, for example, we have a normal range where most people should be. And if you're outside of that range, you either have diabetes or you have some other condition that causes low glucose. So we've developed this panel in a way that works just like blood chemistries. You have a normal range, and if you're outside of that range, for example, your microbes are not fermenting properly, then we know that something is wrong. And then we have other tests to find out what the problem is. Now, to our knowledge, this is the first test that actually can discriminate between a healthy versus unhealthy gut microbiome.
I see. And
And
so this is based on the outputs of the microbiome. It
yes,
doesn't.
what they're doing. I mean,
Yeah.
what that
Yeah.
isn't that what's most important?
Yeah.
What they're doing or what they're making, because that impacts us directly.
Mhm.
Whereas, who makes it doesn't really matter. It's what
Right.
they make.
Yeah. So,
That's
if we just make up a toy example here, there could be some healthy metabolite that I want, above a certain level and below a certain level in, circulating in my body. If I'm getting that from the microbiome, I might be in the healthy range and you might be in the healthy range, but we might have different populations of bacteria that are producing
exactly.
that metabolite.
Yes, exactly.
I see. Can you give us a sense for what this starts to look like in practice? What are some examples here of the functional markers that you're looking at? Are these nutrients that the microbes are producing? What do you actually look at here?
We're looking at a variety of functions that a healthy microbiome must have. So, in essence, what we've created are like the lights or warning lights on your dashboard. Whether your oil has to be changed, or whether your battery is dying, or whether your engine is running too hot. That's what we've created. And so we can pinpoint not only whether you have a dysbiosis, but exactly what's causing that dysbiosis. And that's a big step, because once you know what's wrong and precisely what's causing that problem, then you can fix it in a way that's much more precise than just empirical management.
Mhm. So I mean, what would be some examples of these things?
Okay, so we have several use cases.
Mhm.
There's one guy that developed these symptoms, basically food intolerance. Every time he would eat certain types of food, he would have uncontrollable diarrhea. And this began 10 years ago when his doctor put him on a variety of antibiotics for 18 months for an unclear indication.
Hmm.
Okay, so he developed this problem. It was incapacitating. He was a business executive, had to attend all these board meetings, but before the board meetings, he had to check out where all the bathrooms were, because he would have explosive diarrhea if he ate something. So he had been checked out at 4 major medical centers, and none of them were able to find anything wrong with him. So he finally emailed me and asked whether maybe it's his microbiome. And we said, well, maybe, but we have a test that we can tell you whether or not your microbiome is healthy or not. Turns out his microbiome was severely damaged. He had almost no microbes that were making these fermentation products, particularly something called butyrate, propionate, acetate. These are
Hmm.
really important molecules.
These are short chain fatty acids.
Short chain fatty acids. These are very, very important for immune health as well as metabolic health. And we almost could not detect levels of these short chain fatty acids.
Where would those normally come from?
They come from dietary polysaccharides or fiber.
I see. So sugars or fiber that are then transformed into these short chain fatty acids by
Exactly.
specific types of microbes.
Yeah. We don't have the ability ourselves to do that. We have to, this is one of the major reasons that we have microbes. I mean, this is, if you think about all these ruminants like horses and cows, they require these microbes in order to digest hay and grass. So we have the same thing, except it's at our distal end of our gastrointestinal tract. And coming back to this patient, he had a very damaged microbiome. In fact, when we look further, some of the species, microbial species that are important, that are the main engines for fermentation, were missing. So we decided that, let's try a dietary approach by adding different types of dietary fiber at first, one at a time, see what he could tolerate, and whether or not that was changing his microbiome in a way to become more healthy. Mm-hmm. Were the microbes that he was missing or we could not detect really truly gone or extinguished? Or were they just below the level of detection because they weren't being fed?
So they might be in there a little bit, but they've been sort of boxed out by other microbes potentially. But if you change the diet, if you give them the right fuel, they will start to outcompete those other microbes and gain turf in the gut, and then start producing enough of these short chain fatty acids to potentially fix his problem. So basically what you've told us so far is this guy obviously had a problem in his life that had to do with his gut. You thought it might be due to the gut microbiome. You went in and you sort of diagnosed what the problem could be. He doesn't have short chain fatty acid production at the levels someone should. And this suggests that he doesn't have enough of certain microbes, because we know that there's that connection there between the short chain fatty acids and specific microbes. And so then you did some trial and error to see if you could feed him certain types of food components to fix that issue.
Exactly. So we started with what we call a low FODMAP diet. This is low in fermentable fibers. And then we added groups under the guidance of a dietitian. And over time, and this took over 50 weeks, we could see that what worked and what didn't work. And then we would add more and more. And about mid-course, about 25 or 30 weeks in, we began to see a significant improvement in the health of his gut microbiome. Mm-hmm. And after 50 weeks with this progressive diet, he was feeling better. In fact, he was enjoying life. He could eat more diverse foods. And we can see that his microbiome score was improving to the level where by 50 weeks, he was normal. Mm-hmm. His microbiome was functionally normal.
Mm-hmm. So in your hands, he's working with you, and it takes about a year to fix his problem. You guys are doing all the stuff that you do. You can, go in, measure things, try very specific changes to progressively change, his dietary inputs. What was let's just assume he never met you. What was he eating beforehand? Was he eating just what was not in or what was in his diet that we think was facilitating this problem? And would there have been any way for him to change his diet on his own and fix this? Could he have just started eating a bunch of fiber or something like that, high fiber foods or something, and seen a similar result? Or did he really need to come in and get a doctor prescribed, lab tested dietary shift like this?
So what happened over those 10 years was that he gradually went through an elimination type of approach. If, for example, he ate certain types of food, he would get symptoms, so he would eliminate that from his diet. Mm-hmm. So by the time he contacted me, he was on a very Spartan diet. I have to say that he was only eating like 5 different types of foods.
Wow.
rice and maybe some potatoes and things like that. But he couldn't tolerate anything else.
Okay, so he cut out most stuff because his
He cut it
problems
out.
were that
And
bad. So many things were causing
yeah,
symptoms.
and he still would have an accident when he was eating something outside of that very limited menu. So what we did was, and I don't think he would have been able to do this on his own because
Hmm.
we had a very, very objective test. We were looking at whether we could rebuild his microbiome. And with gradual, and this is why it took 50 weeks, with every couple of weeks, we would add a particular food group and see what it did to his symptoms as well as to his microbiome score. And that helped guide us.
Hmm.
So we knew what worked and what didn't work. And
Hmm.
what worked, we would then add more. And over time, his diet became much more diverse.
Hmm.
His tolerance for the diet was also improving.
Hmm. What was, what were some of the core things that needed to be in his diet to help fix the problem? Was there a core set of foods or food components that were, critical for this to happen that then unlocked his ability to just eat a more normal diet in general?
There were certain types of dietary fiber are um, processed more slowly than others. And so if we found that those types of fibers reduced or didn't cause the buildup of gas and fluid that would cause explosive, uncontrollable diarrhea. So it was really by putting in more complex dietary fiber, which are metabolized more slowly, that gave him the ability to tolerate that and still be able to restore the levels of short chain fatty
Hmm. Hmm.
acids.
Can you talk a little bit about the different types of fiber and what those specific types were that helped him? Because I think a lot of people still think of fiber as it's just fiber, high fiber, low fiber, but there are different types of fiber. What
There
are
are,
these and how do they differ?
oh, they differ tremendously. Without getting into the chemistry, their dietary fibers are basically sugars that are linked by different types of chemical bonds, right? Some of them are very easily hydrolyzed or broken into so that the simple sugars or short chain fatty acids can be formed. If that happens too rapidly, you're going to build up a lot of gas and bloating and diarrhea, right? So, more complex fibers, let's say plant uh, polysaccharides or fiber, have a different type of linkage. And it requires certain types of microbes and enzymes that they have to break it down. But it does it at a rate where you don't develop this big gas
Right,
and
right.
fluid buildup.
So the symptoms, the negative symptoms of that give you some of those waste products that then give you things like diarrhea, that is dependent on the rate, basically on the rate at which microbes break down different fibers. If they do it too quickly, these things build up in your gut a lot, and then you've got to deal with them. We call
Yeah,
that a symptom. That's what manifests in, the things that we're talking about. But if they do it more slowly, this doesn't happen as
that's
much, or,
right.
or potentially
So
at
it's
all.
not just rate; it's how much fiber there is,
Hmm.
right? So uh, it's the rate and the load of carbohydrate or dietary fiber. If you eat a ton of, let's say, Brussels sprouts, and you're going to overwhelm almost any type of microbiome, even when it's healthy. So it's a balance.
Hmm.
Uh, but I think that having a tool that can determine in a very quantitative way whether or not you're out of the normal range, and then through dietary intervention and maybe future microbiome based interventions, you can get it back into the normal range.
Hmm.
That's really what I call microbiome medicine.
Hmm.
This is something that we haven't had before.
It's
and helps us understand the individual's problem and helps us correct it in a more precise way.
mhm. So it sounds like the consequences of eating certain things that contain dietary fiber, the consequences are going to depend on the amount of fiber you consume, the specific types of fibers that you're consuming, as well as the microbial context that fiber is entering. And so all of these things have to be in an appropriate balance to avoid problems. And to the extent that's true, it sounds like what you're saying is you can have too little fiber in your diet, that can cause you problems. It sounds like you can also have too much fiber or too much of the wrong fiber, and that can also cause you problems.
Yes. So what we're doing or will be doing soon is that we're going to be developing a sort of a next generation of medical foods. So we have a system where we can test certain types of dietary fiber on a population of gut microbes and determine what microbes they're affected, what kind of process or rate of formation of, let's say, gas or changes in function of the gut microbiome, so that eventually we have a formulary or sort of a pharmacy of different plant and animal derived food compounds that can be used prescriptively, right? So this is the future of microbiome medicine. You have a diagnostic, you find out what the problem is, then you pick off the shelf that particular ingredient that will correct that problem. It's just like if you have hypertension or diabetes. Once the diagnosis is made, we know how to treat it because we have very specific compounds that can either lower blood pressure or you know control your insulin or glucose levels.
Mhm. Could you maybe give us some examples of common foods that have relatively high fiber content, but that contain different types of fiber, say, and therefore would probably affect someone's gut differently if they ate them? Because again, a lot of people just think fiber is fiber. I should be eating, some amount of fiber, and it doesn't really matter where it comes from. What are examples of different types of fiber that differ in different foods?
So, like beans, right? Beans have a type of fiber that is rapidly hydrolyzed. So, not surprising. You eat a lot of beans, you become very flatulent, and I think that as opposed to other types of dietary fiber that might come from a different type of plant, let's say seaweed is a good example. Seaweed, I think, has a very complex fiber content, and so it causes probably fewer problems than others.
Mhm. So when you say complex fiber content, do you mean that it has a diversity of fibers, each of which is broken down sort of in a different way by different
And its
microbes?
chemical structure
Hmm.
is takes a little more time to break down than, let's say, what you might find in beans.
Hmm.
Now, another example is fermented foods. Okay, uh, I advocate fermented foods because in many ways it represents pre-digested dietary fiber,
Hmm.
right? So this is where the fiber has already been broken down by microbes so that you're getting the final product. You're not getting all the gas and all the other problems that you
Hmm.
associate with just dietary fiber.
Interesting. So, and what are the biggest examples of fermented foods like that?
Uh, you know, uh, like certain types of fermented tofu, and then kefir is another type of fermented food.
So
I
that's like
think they're
fermented
very
dairy.
healthy. So these are like a combination of uh, pre and postbiotics. So
I see. So certain foods can be fermented in your gut, or they can be fermented before they get into your
that's
gut,
right.
and this has important consequences.
mean, if you think about it, fermented foods are very healthy.
Hmm. Hmm.
It's sort of pre-digested. You don't require your own microbes to digest that fiber. It's
Yeah.
already done.
So, and what's interesting about that is, um, it seems like most, or at least many cultures, traditional cultures out there, have some kind of fermented food dish. Whether we're talking about
Yes.
kimchi or fermented soy products or sauerkraut, whatever it is. A lot of cultures have sort of one or two staples in the fermented food category, and perhaps that's not a coincidence. Maybe people just sort of figured out over time through natural trial and error that having some kind of fermented food in the mix was good.
Yeah, I agree. Um, but I think that, diet is underappreciated for what it can do, particularly with the health of the gut microbiome. And this is something that I think represents a opportunity for us to uh, really develop uh, a suite of microbiome based interventions that we know exactly how to use and when, how much, and for how long. So
Hmm. Hmm.
this is, I think, we're moving out of the age of empiricism.
Hmm.
We're actually trying to develop scientific evidence that supports the efficacy of certain of these uh, simple compounds that you can get over the counter.
Yeah. So one thing that's interesting here to me is, so let's take the gentleman that we spoke about that you helped over that 50-week period. What's interesting about this to me is, so his problem came from using antibiotics for an extended period. So he basically carpet bombed his microbiome. This led to his gut issues, and then he had to work with your team of experts for 50 weeks in order to fix that. So on both sides of the equation here, the start of the problem and the solution to the problem, we needed to use, his problem came from an antibiotic drug, something that we invented and developed for a purpose. And then the solution came from this extended 50-week treatment of trial and error that your team had for him. This is also, again, using human brain power and ingenuity to fix a problem that sort of came from human ingenuity, right? The antibiotics that we invented caused the problem, and then the
Yeah.
work that your team did helped fix the problem. What's interesting about all of that to me is, for most of human history, no one had to worry about that, right? No one had to worry about that problem arising. As you said, IBS is a new phenomenon. Things like that have sprung up in recent, in modernity, basically. And then the solution also is, in this case, a 50-week treatment protocol. But for most of human history, people didn't have to worry about any of that. They didn't have
This
to
is
go through 50-week
absolutely
protocols. They
correct.
didn't have to, worry about drugs and all that stuff. And so what is it that, let's talk a little bit more, other than antibiotics, what is it that's causing so many people to have gut issues, to where now we have to invent all of these new protocols and tests and
Yeah.
things that might take weeks and weeks and weeks of time to fix, but for hundreds of thousands of years, none of this was even an issue to begin with.
Yeah. Well, there's good and bad with what's happened over the past couple hundred years. The good thing is, antibiotics certainly have made a difference. There are certain uh, conditions that you require antibiotics. Otherwise, you get these terrible infections. On the other hand, we have created a lot of problems through technology as well as through our diet and lifestyle. Things like Western diets, high in saturated fats and low in fiber, very calorie dense, they have a tremendous impact on our microbiome. We describe something called a Western microbiome. It's low diversity, low fermentation products, very low short chain fatty acids. All these things were not uh, aren't meant to be because that's not how we evolved. We need these things, and because Western diets are, first of all, they're comfort food. People don't want to, once they are on it, they don't want to get off of it. The second thing is that, fast foods are cheap, affordable. And I think that it's hard to change that culture. Yeah. So you have the combination of, Western diets with, the frequent use, often without indication of antibiotics. That really impacts your gut microbiome.
Mm-hmm, mm-hmm. Well,
Now,
a related question I want to ask you, come at this from a different angle, is okay, so for most of human history, people didn't have to do trial and error with their diets to find the exact right type of fiber to eat and the right food and all this. They just sort of ate what was around and didn't have to think about it much. You mentioned earlier that, we were talking about how there's different types of fiber that are metabolized in different ways and at different rates by different microbes. You said, for example, that if you eat a lot of certain beans, this can cause problems because they've got fibers that will be rapidly metabolized, and you will have symptoms that will manifest that you'll actually feel, right? People get gassy and things like that on foods like beans, but other foods don't cause that. Is a simple way to start to assess these things in your own life as a normal person without having to go in to Eugene Chang's lab to get all these tests done is just to see like how much discomfort and waste products is your body making, and eat more or eat less of things so that you're minimizing that stuff.
I suppose, I mean, prior to the sort of new technologies that we have to look at the microbiome, I think that's how people did it, right? If you wanted to lose weight, and you basically went on a, more plant-based diet versus an animal-based diet. And I think that common sense comes into play. We know what's healthy, we know what's unhealthy as well. Our dietitians are always telling us, you just have to eat more fiber and less saturated
Hmm.
fats,
See
and
that
avoid the calorie dense foods.
yeah. That's where we'll probably circle back to this in a different part of the discussion. That's where I start to get a little bit confused because the dietary advice coming from the dietitians and people like that is eat more fiber, eat more fiber, eat more fiber. But previously, you told us that too much fiber can be a problem. There's different types of fiber. So how do we square those things?
Well, I think that the tests and the diagnostics that we're developing can help gauge to what level you and what kind of changes that you can make. Um, the other thing is, why we need tests like this where people can actually see that the change in their diet or lifestyle is actually making a difference is important, because most people sort of lose interest and they become non-compliant on diets or weight reduction type of diet, because they don't see rapid changes, right? It may take months before you see, a loss of a couple of pounds. But in the studies that we have done, so we participated in a uh, program where they teach healthy cooking and selection of better food choices. And we tested these individuals before they entered the program. It's an 8-week program, and then at the end of 8 weeks. And it's remarkable how that instruction and just education has led to improvement of individuals' microbiome health within that 8-week period. But the most important thing is when you show these people their results, it's like an aha moment for them. They become highly motivated. They want to stick with the program. And coming back to this patient that we talked about, that we worked with over 50 weeks. This guy collected 3 stool samples every week for 50 weeks. Why? Because he could see that he was getting better. And he remains highly motivated. He just has been thrilled by the improvement, and knowing that whatever he's doing is making him better.
Mm-hmm, mm-hmm.
So it's that motivational factor, I think, is really important.
Yeah. So I want to talk a little bit more about microbiome depletion and replenishment. So just to riff on this individual again, he had a problem that arose from an 18-month course of antibiotics. And then, he started having all these terrible symptoms, and it took him he had to go on this elimination diet and stop eating most foods until he met you. And then he had to go through this 50-week protocol to slowly add in back certain things to fix his microbiome, to replenish it in the right way. Now, many people have taken antibiotics, and probably some of them have problems that are as severe as this guy. Some of them have problems that are not as severe, and some of them don't have problems at all. So if an antibiotic or something depletes the microbiome, can you talk a little bit about, how likely is it that the microbiome will be replenished and go back to the way it was versus go into a new unhealthy dysbiotic state like the gentleman that you discussed? How much does context and dietary context matter in that replenishment phase in terms of whether the microbiome will go back to the way it was or become healthy or unhealthy when it's reseeded?
Yeah, so that's a good question. And I have a partial answer. So some things that you have to consider that are important for a healthy microbiome are the properties that we call stability and resilience. Right. So what I mean by that is one individual may take antibiotics for, let's say, 2 to 3 weeks. And then once those antibiotics are stopped, his or her microbiome comes back very quickly. So it was very stable and very resilient. It's able to bounce right back, even though the antibiotics were suppressing it for a period of time. Whereas another person who may have a very weak microbiome that is easily perturbed, that person, after the antibiotics, may not recover for a long, long time, and not even to the proper level. And this is a important problem. I mean, we just published a paper on this, where this was a study that was done in mice because it's very hard to do it in humans. But if you put mice on a Western diet and give it a cocktail of antibiotics for a few days, it wipes out the microbiome. I mean, major taxa are missing. Now, if you continue that mouse on a Western diet, it almost never comes back. It stays dysbiotic. And those mice, if they encounter, let's say, a pathogen like salmonella, they get severe infection. Whereas if you had the mouse put back on a more high fiber, less calorie dense, low fat diet, the microbiome comes back immediately. And it comes back with stability and resilience, and it will resist a pathogen like salmonella.
So in other words, if you deplete the microbiome in an animal with an antibiotic and then let it come back by removing the antibiotic, the microbiome that comes back, the composition it has, will depend on the diet the animal is eating. And one diet might enable it to replenish a microbiome in a way that's sort of healthy. And the other one, if you're on a different diet, the microbiome might replenish in a way that's dysbiotic, that makes you more susceptible to infection or gives you some other symptom set.
That's correct.
And I want to talk a little bit about, so that recent study you did, and it had to do with, comparing, diet to microbial transplantation. Maybe a good setup for this is just to give people a sense for microbial transplantation. So to tie this back to the beginning of the discussion, we talked about your concept of the microbiome as an organ. And just like you can have a liver transplant or a heart transplant, you can actually have a microbiome transplant. And there's been a lot of studies out there that do this type of thing, that transplant, say, the microbiome from a healthy animal into an unhealthy one. And they often have pretty dramatic results. Unhealthy animals might get better. It might cure them of, a disease they have, or at least improve it greatly, simply by taking the microbes from another animal and putting them into the one that has a problem. So can you just give us a general sense for microbial transplantation, the types of things that we tend to observe with this, and how we should start to think about it?
Well, the good thing is that, and they're called fecal microbial transplant because they're really from stool or from the colon. This is the last part of the gastrointestinal tract. And by the way, every part of your GI tract has a different microbiome. Okay, so the microbiome that may be in your small intestine is very, very different from the microbiome of your colon. Now, the current state of the art with fecal microbial transplant is that you're basically taking somebody's microbial organ, which is fully matured, interactive, and transplanting it into somebody that has either no microbiome or a damaged microbiome. Okay. And that makes a lot of sense. It's just like an organ transplant. However, the problem with that is that this is really sort of a dark art. We have no idea what's in the donor microbiome. Right? We just assume it's healthy because we take it from a healthy individual, but we don't know whether it's going to fit the needs of the recipient. Okay. The other thing, as I mentioned, is that it only has colonic microbes. These are what we call anaerobes. They cannot tolerate oxygen, and they're very, very different from the microbes that are in your upper part of your gut. So when you do these fecal microbial transplants in some patients, you're only restoring the colonic microbiome. You're not doing anything for your small bowel microbiome. And that's a serious problem. And I think that we have to pay attention to the fact that there is a microbiome in your small intestine that has very different types of functions and benefits that it provides us. So I think that our group is thinking along the lines of, number one, if we're going to develop these microbial transplants in the future, we should know exactly what's in them, right? And we should be able to formulate the microbial transplant to that individual's needs. The third thing is that we have to create, instead of a fecal microbial transplant, we have to create an omni microbial transplant, which includes small and large intestinal microbes, because that way you will the success of restoring the microbiome in the entire GI tract is going to be greater.
I see. How would that work in principle? Would that be using probiotics that are swallowed that sort of go down the tubes that way?
No. Well, first of all, most probiotics probably don't work. They don't engraft. They're mostly from, from milk sources, yogurt and other dairy products. So the one thing is that I mentioned earlier that the human microbiome is very, very specific to human biology and the gut ecosystem. If you put a bovine or a cow microbiome into a human, it will never engraft. Okay, this is why most probiotics, despite the claims, they don't really work. right? So if you're really going to develop an effective uh, microbial transplant or probiotic, it has to come from humans, right? So how do we do that? Well, fecal microbial transplant is one example, but we don't know what's in it. It's the combination from one donor to another is very, very different. And it may not replete the entire GI tract. So, what we're doing now is that we are taking healthy individuals and we're cultivating the individual strains and building up a formulary of these strains so that in the future we can combine them in a way to make an omni microbial transplant that is personalized to that individual's needs.
But how will that microbiome actually get everywhere? How do
Okay,
you get the small
so
intestine part of it into the small
great
intestine?
question. All right. So the best way is to deliver it from above, either by a nasogastric tube or by endoscopy, possibly through these capsules that people are making. The problem with capsules is that you can't control where they're released. And many of them are designed to only be released in the colon. Um, but I think that this is based on our mouse studies. Again, if you give an omni microbial transplant from above, those microbes find their way. They will go to the right place and they will colonize and engraft in the right way and in the right place.
Mm-hmm. What if instead of an invasive method that we come up with like this, if we think about the natural seeding and reseeding of the microbiome that would happen, say, in a newborn child who's just been born, or if someone has their microbiome depleted by antibiotics, say, and then they simply just start eating some food like they would normally, they start eating some particular diet, the microbes will begin growing in them and the microbiome will become replenished. We're not inserting or engineering that microbiome. It's sort of arising organically based on whatever that person is consuming. How should we think about things in that context? So, for example, if someone let's say you're someone out there who has to undergo a pretty significant round of antibiotics because you get sick with something or whatever, and then that's naturally going to affect your microbiome. It's going to be depleted to some large extent. If you then go back to the diet you were eating before versus start eating, say, a new diet at that time, your microbiome will probably grow back differently. How should people think about things like that when they're in a context where the microbiome is depleted and then they're just going to eat some food? Should they pay careful attention to what they're eating in that period after the antibiotics to think about how the microbiome will replenish itself?
Yeah, that's a really good question, and I don't have a complete answer. My uh, feeling, based off of principles of ecology and evolution, as well as some of the studies that we've done in mouse models, is that the more natural that you can make it, the better. What do I mean by that? If you have a forest fire, which burns out everything, what happens is nature goes through a succession of events. So initially, you get the grass growing back, and then you get the bushes, and then sort of the smaller trees, and then the tall trees. That progression is very important because each stage is sort of uh, terraforming the ecosystem for the next stage. So this is exactly what happens when you rebuild somebody's microbiome. It occurs in different stages. There's a pioneer set of species that come in first. They sort of till the soil and they provide the nutrients that the next stage will use. And then that will provide the setting for the third stage. So this pattern of successional recovery is very, very reproducible. And I think it has to happen in that order. Okay. So the study that we did, we found that the best way to make this happen in a natural way that ultimately leads to recovery, almost complete recovery of function, stability, resilience of the microbiome is through diet. So diet is introduced where the pioneer species will metabolize it, produce the molecules that are necessary for the next stage. And so I think that the more natural that way that you can do it is better. You always have to trust nature. Nature knows how to heal itself, and so I advocate, particularly in patients that undergo, you know, a cocktail of antibiotics or prolonged antibiotics, that they maintain this kind of diet that provides the nutrients and substrates that will keep these healthy microbes alive, but give them the opportunity to come back once the antibiotics are stopped.
Mm-hmm. And how readily will the microbiome change simply as in response to a change in diet? So you don't take antibiotics, you don't deplete the entire microbiome, you just start eating a different diet. How
It'll
much
change
of it,
within 24 to 48 hours.
and
That
is
was
that
a study that was done by a very good investigator,
mhm.
Peter Turnbaugh, and it's remarkable how quickly your microbiome can change.
I see. So it can change very quickly. Is it possible but how completely will it change? I guess one way of thinking about this is, let's say someone does have some gut microbiome issue, they're having some symptoms. Can a dietary change be sufficient to change that? Or can the dysbiosis, the dysbiotic microbiome sort of be stuck in place and resilient to a dietary change?
Yeah, I think that that's dependent on who's left. I mean, if you destroyed and extinguished your microbes, your critical microbes that are involved in this successional process, I think the only route is to try to replace them, right? And diet isn't going to be able to replace them. You have to actually replace them with live microbes. Um, Diet is important and probably the best way of repairing a microbiome that has been damaged, but it still is there. So, coming back to this patient that we've worked with for over a year, turns out that, we were able to restore just through diet the functional health of his microbiome. But if you looked at his diversity, we didn't do anything. Basically, he had the same microbes that he started with, at the very beginning. All we were doing was getting more out of the microbes that he had. So the next step was that we're trying to build his diversity up, and so he tried a couple of human recently approved by the FDA defined fecal microbial transplants. Okay, so there are two products that have been FDA approved. And it turned out that the one that was really dependent on what we call spores, very few of those microbes actually engrafted back into them. And I think it's because they were spores, and then they also were not, there are only like 8 different type of strains in that formulation. The future, I think that we can do better. We can develop things that are going to be more diverse and likely to engraft. And by doing that, along with diet, I think we can rapidly rebuild somebody's microbiome.
Mm-hmm. I
Back
want
to
to talk a
full
little
health.
bit more about the interaction between diet and the microbiome and transplantation. So you've got a new paper out called Diet Outperforms Microbial Transplant to Drive Microbiome Recovery in Mice. Can you set this paper up for us? What was sort of the motivation for this, and what were the questions that you were looking to answer with this study?
So the premise is that this is common in the U.S. and other industrialized countries, right? We eat Western diets, and we use very liberally antibiotics. So we wanted to see what the consequences of that combination would be. So the diet actually was designed to be very similar to what uh, Americans eat. This is based off of a population study called NHANES. And so it had the same amount of fat, same macro and micronutrients, very similar to the American diet.
Can you just give people a sense for what that is? When we talk about an American diet, what exactly do we mean in terms of nutrient composition?
So, we typically will eat about 37% of our calories come from fat, right? And in the American diet, a lot of it's from saturated fat, from animal fat. And then there's a certain amount of dietary fiber or lack of it in the American diet. And then protein levels were very similar to what Americans eat. So this was really an American diet.
So, what were those protein levels?
I don't recall specifically. This is a formulation, so it's
Mhm.
a combination of different types of both plant and animal derived proteins. Yeah. So the the consequence of that is that it does decrease the diversity, resilience, and stability of the microbiome. But since the antibiotics were only given over a period of like 3 to 4 days, we found that if you put the mice back on a diet that they're used to, that it has high fiber, very little fat, microbiome comes back very quickly, and they don't. And when we look at what actually causes that, it's this metabolic network that builds up that confers stability as well as resilience. So if you treated those mice or exposed them to salmonella, they were resistant. They either didn't develop the disease or they developed less severe disease.
I see. So the idea here is you've got two groups of mice. Basically, you deplete their microbiome with an antibiotic. One group of mice is on one diet, another group of mice is on another diet. And depending on which diet they're consuming, their microbiome will replenish. It's going to be different in the two groups, and they will be differently susceptible to something like salmonella. One of them will
Yes.
get sicker easier depending on the diet
Yes,
they're eating.
that's right. So, it's a little worrisome because a Western microbiome is less stable, less resilient. And you worry about, whether we're doing ourselves a disfavor because we're going to be very susceptible to, pandemics where a pathogen may sweep through the country and cause a lot of morbidity and mortality.
Hmm. And so, what are some of the questions that you're working on now? You talked about this a little bit before with some of the new profiling that you're doing, but what are some of the questions that you're going to follow up with on this work, or that you're just working on the lab that you're particularly excited about?
Well, I'm excited about a number of things. So we have this one project that we're doing in mice as well as in human volunteers, and that's called having your cake and eating it too. So we concede that it's going to be very difficult for most Americans to uh, stop, with Western type of diets. Um, but what if we supplemented that type of diet with the kind of things that promote the health and propagation of microbes that promote health? things that ferment fiber and so on and so forth. So this study is, if it turns out that, just by dietary supplement, that we can maintain a healthy population of microbes, I think it could be a very practical and immediately usable way of improving health, the microbiome of the general population. Okay. The second thing that I'm very excited about is that I um, am in the process of using a sampling capsule. So this is a capsule that's made by a company called Nimble. Uh, Individual will swallow it, and what happens is that it has a port that collects sample from the time that it passes the stomach to the before it gets, and it closes before it gets to the colon. And you can cultivate healthy microbes from healthy individuals and be able to build a library of small bowel microbes that ultimately will be combined with a library of colonic derived microbes to make this omni microbial transplant that I talked about earlier. And the other advantage is that every strain that we will cultivate, we will know exactly what it does and what it produces. So now we can individualize, customize these formulations for the specific needs of that person. And this is all doable. I think that this is just a matter of time, but eventually, this is, I think, the future. This is how we're going to have precision in microbiome medicine.
Mm-hmm. I want to ask you a little bit more about how Western diets are defined and how we model that in the preclinical research. Different studies use, different formulations of diets and things across the board. And so you really have to look at the specifics there. We've touched on this a little bit already, but when you say Western diet and when you're modeling the Western diet in your studies, what does that mean exactly in terms of nutrient composition? It sounds like, the first bullet point there is it's a relatively high fat diet. So it's something like 35 to 40% of calories from fat, and that a substantial chunk of that fat is saturated fat. Is that part accurate?
That's correct. So we're one of the few groups that actually paid attention to that, right? Most groups that had done fat diet studies use 60% lard. Okay, that's animal derived saturated fats. Well, who eats that kind of level of fat? I doubt that most people do that. Our diet was designed to mimic the composition of the average American diet. And that's well published by the NHANES study. So that's what guided us. And the formulation of the diet, how much protein, where it came from, fat and sugars, was really formulated by a company that we just specified. We wanted 37% saturated fat, so much percent of protein, and certain percentages of micronutrients.
Mm-hmm. Do you know the carbohydrate and the protein content of that diet?
Not off the top of my head, but it is published.
But it was specifically designed to mimic the average American diet. And are there any other salient features of this modeled Western style diet in terms of micronutrients and other things?
I think that we tried to be as close to what the average American eats. And, it's the diet that we have used for the past 15 years. And it's actually proven that to recapitulate some of the, for example, you had done a podcast with a previous student of mine, Suzanne Defcoda. Well, she published this very important, highly cited paper in a journal called Nature, where she showed that on this typical American diet, you promote the emergence of a pathobiont. This is a microbe that has the potential of causing disease by activating immunity and causing inflammation. Well, this pathobiont, which was called Bellophila wadsworthia, was uh, gained an advantage because it uses a um, sulfur, a sulfur that's part of many food groups, particularly in uh, certain types of fats. And the result of that was that in a genetically susceptible mouse that develops colitis, uh, these mice really we increase the incidence and severity of their colitis just by giving this one diet as well as this one pathogen or pathobiont. And I think that became sort of the uh, conceptual model that we now use to think about why certain people get inflammatory bowel diseases and why others do not. Why diet has made such a difference. Why diet could be the cause of why IBD was rare 200 years ago, and now it affects millions of Americans.
Mm-hmm. And when we model Western style diets in the lab, we tend to call them high fat. And they tend to have a good amount of saturated fat, as you said. Proportionally over time, the class of fats that's increased the most in the West are the polyunsaturated fatty acids. Are those contained in any significant degree in the Western style diets that you use in your preclinical work?
So in Suzanne's study, we also looked at polyunsaturated fats, and uh, polyunsaturated fats are just like dietary fiber. They're different types. If you look at, let's say, olive oil versus palmitate, they're very different. They have different actions on the microbiome, and one is considered very healthy, the other is considered not so healthy. So I believe that just saying unsaturated fats are good or bad is probably too generalized.
Yeah. Is a similar thing true for saturated fats? Aren't there multiple types of saturated fats that have different biological effects?
Probably so.
Mm-hmm.
Yeah. I don't have any evidence to prove or disprove that, but I'm guessing that, all fats are not the same.
Right. And do we are we able to take those things into account in the lab? So when we say things like Western diet, high in fat, high in saturated fat, have people matched the exact saturated fatty acid profile to the one that we see people using most often out in the real world?
Well, we have to be careful because mice are not humans.
Mm-hmm.
They one of the difference is they're coprophagic, right? They eat their own feces. Uh, um, so in many ways, they're undergoing constant FMT, fecal microbial transplant. So I would say that we use mice because we can control their genetics, we can control their environment, their diet, and everything, and we can ask questions that we get answers to. But I think that we're accountable for interpreting that data. Ultimately, we have to see if it's relevant to humans. And this is why we almost always do studies initially it's an iterative process. We observe something, come up with a hypothesis based off of human observation, and then we take it into a mouse model, and then we try to find out what the cause drivers and mediators are. But then we have to go back to the human to determine whether that is relevant. And I think that is absolutely necessary. Otherwise, you get misled.
Mm-hmm. Um, Is there anything that you want to reiterate from our discussion or any final thoughts you want to leave people with about the microbiome or any of the things that we talked about?
Yeah. So, why I am so interested in the microbiome is that unlike other vital organs of the body, it's easily modified. Okay. So I'm doing a study right now that's funded through the NIH, includes 5 major academic centers in the United States to look at the gut-brain axis in patients with Parkinson's disease. There's a large amount of evidence that disruption of the normal gut-brain axis can lead to not only Parkinson's, but other neurodegenerative disorders. So this study is a consortium of these 5 institutions. We're part of it. Our role is really to look at the microbiome, and dysbiosis has been associated with patients with Parkinson's disease. So when I think about it, right now there's no treatment. I mean, we can treat symptoms, but we can't affect the natural course of Parkinson's disease, which is a sad statement. But if the microbiome had a role, then I think that this is a modifiable driver, and maybe we can mitigate the risk as well as development of Parkinson's disease.
All right. Well, Dr. Eugene Chang, thank you very much for your time.
It's a pleasure. Thanks, Nick.
Visit mindandmatter.substack.com to find all of my content. If you like to read like I do, then you should check out the ReadWise app. It's totally free to download, and if you go through the link in the episode description or on my support page, you can get a free 60-day trial for 0 cost. ReadWise is a really cool app that allows you to import digital highlights from over 30 different platforms such as Kindle, iBooks, PDFs, and more. It makes it really easy for you to review and find and share excerpts and quotes from whatever it is that you're reading, whether it's PDFs of scientific papers or books that you're reading on your Kindle, and you can even save and use highlights from physical books. I read a lot of physical books before bed, for example, or when I'm traveling, and the ReadWise app actually integrates with the camera on your phone so that you can take a photo of a page that you're reading, highlight a specific chunk of text from that page, and export it to the app so that you can reference it or find it or share it later. They even allow you to export really cool looking graphics of some of the highlights that you have in the books or the papers you're reading so that you can share them in an engaging way. So check out the ReadWise app today if you are a reader and you like to reference and organize what you read. Go through the link in the episode description or on my support page or in my free weekly newsletter at mindandmatter.substack.com and try the ReadWise app 60 days free today.